Our next speaker, Shohat-Tal, is going to present the FMR1 gene in human reproduction. She's a postdoctoral associate, Department of OBGYN at Yale, and assistant visiting scientist at the Center for Human Reproduction.
Thank you, the organizers. The title of my talk is the FMR1 gene in human reproduction. You see here the X chromosome and the fragile site. And what gives rise to this fragile site is the CGG repeat in the FMR1 gene. If it repeats more than 200 times, it gives this dangling appearance to a piece of the chromosome. Now, CGG repeats with lower repeat numbers can actually is actually associated with a decline in ovarian reserve, and this is going to be the topic of today's talk. I have no conflict of interest to declare. So the Fragile X mental retardation gene has a CGG repeat region in the 5' untranslated region. So this is a region that is not in the open reading frame of the gene, so it does not affect the coding region. It doesn't affect the amino acid of the sequence of the protein. It is highly polymorphic. It repeats as a different amount of times in different individuals. And if we look at the frequency of CGG repeats in the general population, what we will see is a very narrow peak between 29 and 30 repeats. Now, this repeat, suggesting that the majority of individuals have these repeats numbers, is suggestive of very strong evolutionary conservation, such that deviation from this range in either direction may have some substantial reproductive consequences. So, currently, CGG repeats in the FMR1 gene are divided into at least 4 classes. So there's the normal range that is has up to 55, well, actually up to 45 repeats. The gray zone alleles, which have from 45 to 54 repeats. The pre-mutation range, which is between 55 to 199 repeats. And the full mutation range is associated with carriage of one allele of at least more than 200 repeats. So what we currently know is that the pre-mutation range is associated with elevated mRNA expression of the FMR1 gene. This elevated, the CGG repeats are actually responsible for creating these hairpins in the mRNA of the FMR1 gene. And these are pretty stable and resistant to translation. And the result is that there is a reduction in the amount of FMR protein being produced. These structures also sequester CGG binding proteins that are necessary for the translation of other proteins within the cell. And in neurons, it was shown that this increase in mRNA results in the formation of inclusions or these plaques that are often associated with Fragile X tremor ataxia syndrome, which is more common in males. And in females, elevated FMR1 mRNA levels are associated with premature ovarian failure. So this is known as the gain-of-function toxicity hypothesis. So it is not So it is not actually believed that it is the reduction in proteins levels that are associated with these phenotypes, and that is because premutation alleles have the potential in one generation to expand to the full mutation, and this is where during crossing over of meiosis, during germline transmission, the alleles less than 200, between 55 and 200, can expand to well above 200. And this results in widespread hypermethylation in the promoter region of the gene, and resulting in virtually no Fragile X mental retardation protein being produced from this allele. Now, women carriers of the full mutation have no ovarian phenotypes. So it is believed that it is not the protein or reduction in protein levels that are associated with POF, but rather it is the increase in abnormal mRNA levels, abnormal that have these weird hairpin structures. Now, what you will see is that the CGG repeats in blue can be interspersed by AGG interruptions. So it is believed that these AGG interruptions actually prevent or break the formation of these hairpin structures, and they significantly reduce the risk of the expansion of a premutation allele to the full mutation. So AGG interruptions were shown to actually decrease the risk of expansion of the mutation allele. The more interruptions you have, the more the risk of expansion is moderated. Maternal age goes in the opposite direction, so increasing maternal age was shown to increase the risk of transmission. And that is why women carriers of the premutation are often advised to undergo PGD to preclude transfer of embryos with the full mutation. So we mentioned that it is not necessarily the protein that is responsible for the ovarian phenotypes, but just to mention what is the function of the FMR protein, well, it is a translation repressor. So it binds mRNAs and prevents and actually regulates their translation. There has to be some checks and balances in the cell. And you can see that in the case of Fragile X syndrome, this loss of FMRP leads to abnormal protein synthesis, resulting in increased expressions of synaptic proteins, leading to abnormal synaptic function. So now I'm going to talk about some ovarian phenotypes associated with the premutation and the gray zone. So Fragile X associated primary ovarian insufficiency is reported in 20 to 25% of women with premutations. Conversely, 2 to 15% of women with POI carry the premutation. POI is increased up to 30-fold relative to the general population in carriers. And Fragile X POI is associated with irregular menses, decreased fertility, elevated FSH, and early menopause. In fact, women carriers of the premutation have on average enter menopause on average 5 years earlier than their siblings without the premutation. And the association between CGG repeat numbers and POF is not linear, in that CGG repeats of 80 to 100 have the highest risk of POF. The gray zone alleles, again, these are repeat lengths of 45 to 54. So there's rarely any expansion to the full mutation in one generation. If there is, it doesn't go above 60 repeats. But the potential to expand to a full mutation is higher over 2 or more generations. But if the permutation is inherited from the father, then the risk is completely minimized for some reason. There is some evidence to suggest that the incidence of these alleles is higher in women with POF or DOR, but the results are conflicting, and it is presumed that this is due to different populations that were analyzed and may be race dependent. And finally, it is associated with slightly increased FMR1 mRNA levels. So, what are some manifestations of the premutations, some ovarian manifestations? So, there are some insights from mouse models. The growth development of the ovary and the establishment of the primordial follicle pool is normal in premutation mice. However, they show a faster loss of follicle of all classes with age. And this is concomitant with increased rate of follicular atresia, reduced phosphorylation of AKT and mTOR, which are required for granulosa cell proliferation and survival. Adult premutation ovaries are significantly smaller. They are significantly smaller. And they have fewer granulosa cells per follicle, lower FMRP levels, and elevated mRNA levels at all ages. And premutation mice have significantly elevated FSH levels, reduced LH during adulthood, and decreased of number of pups per liter, which is consistent with POF. So now I highlighted this faster loss of follicles of all classes because I'm actually going to bring up Drosophila models in which FMR was shown to be required for suppressing primordial germ cell differentiation. These two things are they are reminiscent of the AMH phenotype, the AMH null phenotype, and which I will show soon. But they also suggest a role for FMRP in inhibiting primordial follicle recruitment. So there are some insights from AMR null mice. And this is from 1999, but Dr. Sen today will present much newer evidence or much newer models. In ovaries from 4-month-old AMH null mice, there are significantly fewer primordial follicles, accompanied by an increase in the number of small growing follicles. Whereas in ovaries of older mice, more aged mice, 13 months old, they have virtually no primordial follicles. Whereas in females that are heterozygous for the mule mutation, the number of follicles fell in between the numbers found in wild-type and AMH null mice. This suggests that AMH, in a dose-dependent manner, is responsible for, inhibits the recruitment of primordial follicles, such that in its absence, there is premature depletion of primordial follicles. And this is the current model of the role of AMH in inhibition of follicular recruitment. And evidence with FMR suggests that it may also be functioning to prevent the recruitment of primordial follicles. So it can act independently or it can act together with AMH. So because of the some evidence suggesting common roles for both FMR and AMH, and because they both seem to be they both seem to be have associations with race. If you look at the at GCC counts in well, this is patients with infertility that are stratified by race. This is a box and whisker plot. You see that in the African American population, there is a higher frequency of alleles with lower CGG repeats, whereas in the Asian population, there is higher frequency of CGG repeats with higher numbers. So this suggests an association between race and CGG counts. The median of the population is the same. It's just that the frequency of alleles above and below the normal range is different in different races. And we also know that there is an association between race and AMH levels. So Dr. Seifer demonstrated that compared to white women, AMH levels are lower among Black, significantly lower among Black, and lower in the Hispanic population relative to Caucasians, wage adjustment for age, BMI, and smoking status. So FMR and AMH definitely have some common features, and AMH levels are known to be about 46% reduced in premutation carriers relative to age-matched controls. So the question is there a relationship between AMH dynamics and FMR genotype in women carrying intermediate and normal alleles? So we looked at this question in the literature, and we looked over more than a dozen papers, and we saw that some papers demonstrate a relationship, whereas others do not. And we tried to look at what's in common to reports suggesting of no association compared to those suggesting that there is an association between FMR repeats and AMH levels in the normal range. So the studies finding no association between FMR genotype and AMH levels include otherwise fertile women. So that is oocyte donors or women with repeated pregnancy losses. Some call it spontaneous abortions, but they don't have an apparent ovarian dysfunction. So that's why I say otherwise fertile women. They don't have any ovarian dysfunction that is apparent. And no stratification is provided in the study for age or the number of CGG repeats. And conversely, in studies that do find an associations, they perform some stratifications. So this is a study that's stratified by age. So Gleischer's group looked at AMH levels in carriers of different CGG repeats in allele 2, which is the longer repeat allele. And they found that in women carriers of shorter repeats women younger than 38, there is a very clear association in that shorter CGG repeats have higher AMH levels relative to women with repeats that are higher than 35. And then there are studies that stratified by CGG repeat numbers in different ways. So they either looked at a specific cutoff or a specific type of repeats. So when I say number, I mean they stratified by CGG repeats below 35 or above 35 repeats. So Pastore's group showed that the rate of decline of AMH in carriers of more than 35 repeats is significantly faster than that of carriers of less than 35 repeats. And the graphs intersect around the age of 36, such that in women with over 35 repeats have higher AMH levels at younger ages, whereas at older ages they have significantly declined AMH levels. The other way of looking at repeat numbers is by stratifying by normal, heterozygotes, and homozygotes. So Gleischer's group demonstrated that using a box and whisker plots, that the normal range for women with infertility is between 26 and 34 repeats. So they called that the normal range. Women carriers of more than repeats above or below this range, so repeats with less than 26, so alleles with less than 26 repeats or above 34 repeats are considered heterozygotes in that they have one normal and one above or below that range. And then homozygotes are two alleles that are above or below that range. And they showed that homozygotes have, all across the board, significantly lower AMH levels, whereas these homozygotes are relatively rare in the population, whereas heterozygotes, which are more common, have less of steep decline in AMH, such that at younger ages their AMH is lower, but at older ages their AMH is higher than women with both alleles in the normal range. And Gleischer suggested that this can be suggestive of preservation of fertility into older ages in women carriers of one allele outside of the range. And this is one last way that CGG alleles were stratified. So it was, so Gleischer's group again looked at oocyte donors. But this analysis is cross-sectional. It is not longitudinal like the other ones. So they looked at oocyte donors over a period of 4 years since they entered the donor program at CHR. And they looked at women carriers of at least one low allele, less than 26 repeats, as compared to all the other genotypes. So normal and carriers of one high allele, one high repeat allele. And they noticed that they have a much steeper decline of AMH over the range of 4 years that they were in the program. And I believe they actually started counseling these donors about the potential of their AMH to, or ovarian reserve to more quickly deplete. And here's another example of stratification by both CGG repeat numbers and age. So this is based on the same data as in the previous figure.
And again, thank you very much for your attention. And
both
for your
stratifications allow you to see that specifically between the ages of 30 to 40, women with normal CGG repeats between the range of 26 to 34, they have the steepest decline in AMH, whereas women with alleles outside of the range have a more moderate decline and a less steep one. So this suggests a model by which distinct FMR genotypes differentially regulate the rate of decline of AMH. Now, how this happens has to be investigated. It could be proposed that, knowing that FMR premutations are associated with decreased number of granulosa cells, this could be the cause of the rate of decline of AMH. Perhaps there are less severe phenotypes even within the normal range. We also know that in Drosophila, FMR1 plays a role in the microRNA pathway. So potentially it may bind the AMH mRNA and prevent its translation being a translation repressor. But this has to be looked into more thoroughly. And some conclusions are that CGG repeat numbers below the premutation range, they already denote a risk for premature ovarian senescence. FMR testing in young women may allow predictions of the risk towards diminished ovarian reserve and can be predictive of the rate of decline even before the onset of ovarian aging. However, additional longitudinal studies are needed cross-sectional studies, are required to provide a more accurate picture of the association between CGG repeat numbers and AMH levels. And the potential contribution of AGG interruptions to the dynamics of AMH decline, as well as the molecular mechanisms underlying this association, should be investigated. And I would like to acknowledge Center for Human Reproduction, Dr. Gleicher, Barad, and Dr. Kushnir. Thank you.
Thanks, Anne.